Hi, this series of lectures is about genetic testing in the genome era. It will be an introduction that includes the most important concepts in genetics and genome biology. I'm your instructor. My name is Arend Sidow. I'm a professor of pathology and genetics at the Stanford University School of Medicine. I'm a PhD. My PhD I received at the University of California in Berkeley in genetics. My initial work centered on molecular evolution, which is the study of how DNA changes over geological time amongst species. As a postdoctoral fellow, I worked on mouse genomics, trying to understand certain aspects of limb development. And since I became faculty at Stanford, we have been working on molecular evolution, gene regulatory mechanisms, and most recently on cancer evolution and regulation. So this course is about genetic testing. It all starts with DNA, of course. DNA contains the instruction that is necessary for an organism to become a living, breathing being. The physical conversion of that information that's encoded in the DNA into an organism involves an incredibly complex set of chemical reactions, really, that build cell to cell into an organism. When the instructions in the DNA are impaired in some way, the biology that builds the organism goes awry potentially, and the outcome is not within the norm: disease, malformations, perhaps even death. Geneticists refer to this as genotype, phenotype. The genotype is the information in the DNA, genetic variation. The phenotype is the outcome. What genetic testing is supposed to do is test the genotype so that one can predict the phenotype. And so in that sense, genetic testing is really genetic prediction. Conceptually, this is really not all that different from, let's say, a blood lipid test. You test, and the results that you get are fairly precise measurements of your blood lipid levels. In terms of what that means for your biology, that is fundamentally a prediction and a risk assessment. If you have elevated triglycerides, it doesn't mean that you are for sure going to be having heart disease. It means that you're at a certain elevated risk of heart disease. And genetic testing is really no different. There are very, very few genetic tests which, when run, determine what you're going to be like in the future. Most genetic tests are really about risk prediction. Sometimes, depending on exactly what is being tested, a highly elevated risk for something, but sometimes not very highly elevated risk at all. So let me tell you how I'm going to do this course. The relationship between genotype and phenotype, of course, rests on scientific progress, where study after study after study accumulates knowledge. Now, biology is so complex that there are really far too many details to cover in a short course like this, but we need to cover some important concepts that underlie the fundamentals behind genetic testing. So concepts accumulate over time as well, but there are far fewer of them, and it is much easier to understand than all of the details here. So what I'm going to do in this course is I'm going to focus on the concepts, and then I'm going to illustrate the concepts with specific individual details that bring life to these concepts so that you have an anchor upon which you can kind of imagine things and understand things when you're presented with these concepts in the future. The first conceptual framework that I would like to give is from the perspective of a human being, the functional organization of nature, of life, actually nature. Physics, and we have the disciplines here. Physics deals with atoms and subatomic particles. When atoms interact with each other and form bonds, we have chemistry. When molecules that are formed by those bonds between atoms interact with one another in more and more complex ways, and when cells are built, we have cell biology. Cell biology, of course, is the study of exactly how cells behave, and cells themselves make up organisms and organs, which we study using physiology. And then at a yet higher level of organization, we of course have culture, tribes, and societies, and so forth. The point I would like to make here is that as soon as biology gets into the picture, there is an orthogonal axis that develops, and that has to do with the inheritance of information. Organisms reproduce, live and reproduce, and over the generations. And quite separate from the function of the organism, the information that gets transmitted from generation to generation, it has to be transmitted across the generations. And so as a consequence, we have several levels of genetics, if you will. There's transmission genetics, which is the study of how a trait is inherited from one generation to the next. Following that, there's population genetics, which is all about how genetic variation is distributed within the interbreeding population. And populations over time evolve into species. So these two fundamental axes, if you will, are what this first part of the course is going to be about. So that is the first concept that I wanted to get across. The second one has to do with scale, because I think it's quite important for us to understand when we talk about biology at what scales it happens. And what I've drawn here is a patch of skin with hair. That's across approximately 2 by 2 centimeters across. And of course, we all have a good understanding of the kinds of scale that we interact with on a daily basis, the kinds that we see and touch and can feel. And then I think most of us have probably also had at least some high school biology or chemistry, and so we're familiar with the scale of atoms. But if you compare these two here, this is actually a magnification of approximately 50 million fold. And in between these is where all of biology happens. So let's illustrate that by zooming in, magnifying a few orders of magnitude. So a 10-fold magnification of this patch of hair. You will see hairs and their follicles embedded in skin, and this is approximately 2 by 2 millimeters here. Zooming in another 10-fold, you can see the hair follicle, and you're beginning to see in this slightly amateurish drawing that there are individual cells that surround the hair follicle. So there are cells that form a sebaceous gland that make the oils that cover the hair. There are cells that are support cells, if you will. Here's a little bit of a muscle cell that comes in, because when you're cold and your hair stands on end, that's when that muscle pulls on that hair. Zooming in another 10-fold, we get to the level of cells themselves. So this is 1 centimeter. This is 10 millionths of a meter. And I want to pause here for one moment and explain the scale of cells, because cells are the fundamental unit of biology. And I think we want to have a fairly good quantitative understanding of that scale right there. So let's do a little bit of math here, or at least a few numbers. A cell diameter is approximately 12 micrometers. Okay. So now the question is, how many cells do you think are in the tip of your thumb, say? The tip of your thumb is approximately 1 cubic centimeter. And without going through the conversion of this one-dimensional metric to this three-dimensional metric, I'm just going to tell you that 1 cubic centimeter of tissue where the cells are densely packed is no less than 10 to the 9 cells, or a billion cells. A cubic millimeter which is, you know, a mighty small bit that you might just barely see if your eyesight is as bad as mine is nowadays, is still 1 million cells. Now, a cubic centimeter, 1 billion cells. Let's talk about your brain. Your brain is approximately 1.5 liters. 1.5 liters is approximately 1.5 trillion cells. And you, the whole human being, is approximately 100 trillion cells. Okay. Now, cells being the fundamental unit of biology, that makes it really quite staggering, right? That these numbers contribute to your body and contribute to all of your function of your body. 100 trillion cells. Now, before we get too carried away with the complexity of it all, we're going to break it down further and look at individual cell biology. So, like I said, this is a cell here, and it's a little bit hard to see, but I just want to point out this is the nucleus here. It's stained with a certain dye that shows DNA, and that's why it's glowing blue. These pieces here are intracellular structures, and in between is the intercellular space. Now, these cells here are grown in culture, so they are not abutted right next to each other, but cultured cells make pretty pictures. That's why I'm showing you that. Zooming in tenfold further, you see subcellular structures, and then zooming in a further tenfold, you begin to see individual little blobs here that are protein molecules. And I want to just point out real quick, for the first part of the course, I'm going to be driving towards proteins because proteins are the machines of the cell. And as you probably know, DNA contains the information that makes proteins. We're going to get into that later, but basically the whole focus of the first part is going to be cells and the machines within them, the proteins. So zooming in about 50-fold here, or 100-fold, this is a gigantic protein machine. Each one of these little balls is an atom. They're arranged in specific configurations so that the protein can do its work. And then zooming in further, we're at the atomic level, where this molecule here consists of perhaps 100 atoms. And now we have a sense of, with 50 million-fold magnification of the scale of biology. Again, the main sort of focal points are the cells and proteins and how they're organized within the cell and how they function. Okay. Now, if you wanted to have a analogy, sort of a visceral understanding, let's say that a protein is the size of a machine, of a human-sized machine. At this scale, you'd have a factory, and the cell would be like a town. With one important difference, when we envision ourselves within a town, what we think of is quite two-dimensional, really. You look at a map, it's a two-dimensional map. And there's a lot of space, right? There's a lot of airspace in the town. But in the body, cells are packed tightly against one another. And so you have really, first of all, a factory where the machines are really, really densely packed. And then you have factory after factory after factory stacked in 3 dimensions, and the town is an entire gigantic town of buildings closely abutting each other in all 3 dimensions. So that's where these kinds of scale analogies kind of break down. Suffice it to say, though, that as you might imagine, a protein machine is only a tiny, tiny little part of a town. Okay, so with these concepts in mind, I wanted to just briefly go over the course content. There's going to be introductory content, and then there's going to be specific content. Of course, what I just talked about is already some introductory content. What we need to understand is some basics of biology. So I'm going to be focusing on cell biology and the machinery, how cells are organized and how they work, how the proteins within them work. And then in a second lecture, we're going to go into the genetics, the information, and how the machinery is encoded in the genome and how the genome is inherited, how the information is transmitted. And I remind you of this slide here, where I was making the point that we have the functional part, which is the cell biology and protein function, which is really orthogonal to the genetics. Here's the function, here's the genetics. And just to highlight that once more, the inheritance of a trait follows certain rules. And it really doesn't matter what the trait is. If you've heard of an autosomal dominant disease, for example, it might be a disease of muscle, or it might be a disease of brain. Completely different functions, but the same concept, the same kind of inheritance. Similarly, you might have cell biological processes that are impinged upon by genetic variation, but the genetic variation might behave in completely different ways. So these two are really fundamentally different interacting issues. Okay, so cell biology, genetics, and then we're going to delve in a little deeper on genetic variation and disease. We're going to ask about variation and diversity and about phenotypes. And finally, there is the specific content where we're going to talk about DNA sequencing and related approaches, i.e. how genetic tests are being done physically in the lab. And this is where we're going to get into the weeds a little bit with details. And then, of course, genetic tests themselves. So that's the outline. And I will see you again when we talk about cell biology in the next lecture.